Hello everyone, and welcome to this third talk of the OHBM 2020 Symposium, Charting Brain Variability in Health and Disease Using Normative Models. In this talk, we will switch gear a bit and kind of go away from looking how we can use normative modeling to predict individualized deviation in individuals with mental ill health, and look more in how we can use normative modeling in a more technical way to look at site differences in multi-site neuroimaging studies. And the title of my talk is Accommodating Site Variation in Neuroimaging Data Using Normative and Hierarchical Bayesian Models. And with that, let's jump right in. Just a quick introduction. My name is Johanna Bayer. I'm an almost PhD, so I'm a finishing PhD candidate at the University of Melbourne. And the study I'm going to present was supervised by my primary supervisor, Leanna Schmall, also at the University of Melbourne. And another supervisor for this project, or like my co-supervisor for this project, was Andre Marquand, who is on this symposium. And I also got support and feedback from Richard Dinga, who is a postdoc with Andre, or was a postdoc with Andre in the Netherlands. Yeah, so the background for this study is side effects in pooled neuroimaging data sets. So in neuroimaging, there exists this trend to pool neuroimaging data across studies to create large neuroimaging data set. That's with the obvious benefit that, neuroimaging is costly. And we can have more data if we just gather data and put it together in one data set. However, the data sets that are created in such a way are often affected by acquisition site related batch effects, also called side effects. And these effects stem from the fact that different studies use, for example, different scanner software, different field strength, different scanners. And these effects, differences affect the resulting neuroimage. In addition, there's another issue, which is that these scanner effects are often correlated with biological variation or other variation of interests that differs between sites. For example, some sites have an older age range of individuals, other sites have more women, and this kind of interacts with these side effects. And the issue of side effects has been recognized by the field, which led to the development of statistical methods that can harmonize for those effects. So one prominent method is COMBAT, which many of you might have heard of. And then another nonlinear extension of COMBAT has been developed by Pomponio just recently. However, it has been shown that these methods cannot accommodate for the interaction between side effects of variation and side effects and variation of interest, and they tend to remove too much variation of interest, so too much biological variation. And Nygaard showed already in 2016 that the removal of too much variation of interest can lead to overconfident results in downstream analysis. If you're not familiar with the Nygaard paper, I would recommend checking it out. It has been a bit overlooked, but it shows that really nicely. So what I've plotted here at the bottom of the slide is side effects as they can be found in the ABIDE dataset. The ABIDE dataset is a dataset containing healthy controls and individuals with autism spectrum disorder. And I took the free surface parcellation data, so cortical thickness of 34 brain regions. And so what I did is each box plot that you see here is basically the 34 brain regions, distribution of these 34 brain regions, cortical thickness. And then what I did is I grouped the data by site, so that's the colors, and within site I ordered it by age. And you see that in general, cortical thickness decreases with age, but you also see that there are huge shifts between the sites. So that is basically side effects. So as we've already heard, normative modeling. Normative models map the variation from one dimension onto another, allowing to express each individual score as a percentile or Z-score of deviation from the norm based on a predictor of choice. Also normative modeling, if it's implemented in a certain way, which has been nicely developed by André Marquand primarily, allows for the separation of different sources of variance. And these two features of normative modeling can now be used to correct for side effects. Kind of the most important sentence, or the most important part of the sentence, is that normative modeling maps variation onto a predictor of choice. So instead of just normalizing for age and sex, we could now add site as our predictor in a normative modeling. And what we would then get as z-scores is basically z-scores that are not only normalized for age and sex or whatever other biological variable that we're interested in, but also for site. And yeah, I've just, I guess you've seen this equation like a couple of times now, plotted here how we get the z-scores. So basically, the normative modelists that we work with have like, basically, they look at the difference between the value predicted by the z-score, which is y hat, and then the observed value of an individual, and then divided by several sources of variation. So in this case, we have like a sigma E, which is statistical noise, and then sigma S, which is meaningful biological variation. Yeah, like I said, my idea was to use this like site as a predictor in a normative model, and then see how good this model could accommodate for side effects. And so what I did is I used, like I said, data from the Bayer dataset. The Bayer dataset contains 569 healthy controls and 482 individuals with autism. And I used the cortical thickness data from this dataset, the free surface population data, for model development and testing. And most importantly, the Bayer dataset contains data from 20 different sites. So what I did to test the models that I wanted to develop is, you see that on the right-hand side, as you see my testing pipeline. So I took the Bayer sample, I divided it into an autism branch and a healthy control branch. And the features, like I said, I was looking at as bilateral cortical thickness measures of 35 regions, so 34 regions, and then an average across these 34 regions, so 35 points. And then I divided the training data set into the healthy controls into a training set, which was 70% of the data, and a test set of 30% of the healthy control data. This division kind of allowed me to check how well the model works in general. And then I also tested the model that I trained in the training data set onto the autism branch of the data set. This allows me to see how well the model performs in a data set that contains other variation of interest, for example, disease status. Yeah. And yeah, from all these three branches, I get z-scores that I'm then looking at. Yeah, so the models that I developed, I developed two different types of normative models, and the way I implemented them is in the form of a hierarchical Bayesian model. The two types of hierarchical Bayesian models that I developed is a linear version, where I had a linear association between the predictors and the Bayesian variables, and a nonlinear version that included a Gaussian process, which allowed a nonlinear association between the predictors and the variables. Yeah, and in order to see how well my normative models actually perform in accommodating for site, I compared the performance of those models to different other side effect correction models that are out there, or methods. The first one is residualization, so just regressing outside. And then I looked at the combat model. So the combat model allows to run just combat on the data, but it also allows to preserve certain variables of interest. For example, age and sex is very common. And then I also applied combat gum because that's this nonlinear extension by Pomponio. So the problem in comparing these two types of models, like the normative and the comparison models, is that the output of these two types of models is very different. From the normative models, I get z-scores right away, but from the comparison models, I just get adjusted cortical thickness scores, basically adjusted for sight. So what I did with these scores is basically pipe them through a very simple normative model, basically just a conversion to z-scores, in order to have also z-scores at the end, in order to compare the two. And that's why I call this two-stage models, because I have these two stages, whereas the normative models is just one stage. I get z-scores right away. And then I looked at how these two types of models perform in terms of model performance. I have different parameters of model performance. The Pearson correlation coefficient rho, the standardized root mean squared error, the log likelihood, and the explained variance. And then I have the mean standardized log loss and the predicted variance. And the reason why I group these 6 variables in the way I group them here is because the first 4 only look at the basically how much the predicted score by the model differs from the observed value, whereas the mean standardized log loss and the predicted variance also look at how well the models perform in terms of predicting variance. And so results, first in the application of the trained model to the healthy controls test set. So just to say it right away, the hierarchical Bayesian linear model and the hierarchical Bayesian Gaussian process model outperformed all other models with respect to these linear model comparison methods. So that just look at the deviation between the observed and the predicted value. And I show you plots for the 2 measures that are more interesting because they look at the variance. So the first one is the mean standardized log loss. The mean standardized log loss basically tells you how much your model performs better than just the mean of the training set in terms of predicting data. And for the MSLL, values that are below 0 are better. That means that the model performs better than the mean of the training set. And what you see here is for the hierarchical Bayesian Gaussian process model and the hierarchical Bayesian linear model, that almost all 35 regions show values below 0. The combat GAM and the combat model perform well for some regions, but not for all regions. And you see that the covariates and the residual models perform very poorly. What I then did also is look at the predicted variance. So just plot the predicted variance, the variance predicted by the model, or like the points predicted by the model across the, against the observed values. And you see like on the right-hand side, like this plot at the top, that the hierarchical Gaussian process model and hierarchical Bayesian linear model, the points lie on the regression line between the two, the observed and the predicted values. Whereas for the combat models and also for the residual models, you see that if you look at the predicted values, that the range is shrinked. So we have a shrinkage of variance and the points are squashed together. And we interpret this in a way that these models take out too much variation that would actually be meaningful. So yeah, you have the reduction of variance here. It's just too much being taken out. And then as in the second step, I applied my hierarchical Bayesian linear model and the hierarchical Bayesian Gaussian process model to the clinical branch of the Bayer dataset. And so what I did here is just basically plot the percentage of z-scores, like the predicted z-scores that are in an extreme range on the brain region that they are in, basically. So the way the normative model is defined is that we should have around 5% of extreme values for each brain region, basically. And that's just the z-scores below and above 1.96, basically on each end, basically because that's how the Gaussian curve is defined. And for the control test set, like at the top, you see that the threshold is at 5%, that there's only very few regions where it's actually a little bit above 5%. And if you calculate the average, it's pretty much around 5%. Whereas for the autism test set, the average of extreme Z scores per region is around 7.5%. The way we ran this study actually doesn't really allow us to make too many statements about autism, because we used bilateral data, and autism is shows laterality effects. But the point of the study also wasn't to investigate autism. It was more to test whether this normative model can accommodate for side effects. So basically, what we conclude from this is that the model is able to retain variation or like differences between the clinical test set and the control test set, which shows that it deals with variation in a meaningful way. And then the next thing I looked into is whether site can still be predicted from after correction with the models. And you see in the top row is the raw data on the left-hand side for the training set, the test controls, and then the autism data set. And you see that there's a lot of variation. So you see like the sides on the y-axis, and then the Z scores on the x-axis, and you see that there's a lot of variation. After the application with the hierarchical Bayesian linear model, you see that this variation is quite reduced, especially for the training set and the autism test set, and the same for the hierarchical Bayesian Gaussian process model at the bottom row. I also have to say that in this study, there was no difference between the linear and the nonlinear model of the normative models. And we think that is because we used cortical thickness, and cortical thickness doesn't show too many nonlinear effects with age. So what we conclude from this study is that including side as a predictor in a normative model is an alternative to common side effect correction methods. In addition, in our study, those methods the common side effect correction methods led to an undesirable shrinkage of variance due to the removal of variation of interest. Normative modeling, on the other hand, retained variation of interest in an autism sample. And we used a Gaussian process regression model for the nonlinear modeling of effects. However, this method is very costly. So for another application in the Enigma MDD dataset, I've developed a splines version of the model, which is way faster and allows an application to even larger data sets. If you're interested in this study that I just presented, I put a QR code here to a preprint, and then the QR code at the bottom is another paper that I just put out on SciArchive, basically comparing different side effect correction methods. The top paper is a bit opinionized because it's basically the study and the results. The bottom paper should be more objective, comparing advantages and disadvantages of different methods, including combat, deep learning, neuroharmonize, and normative modeling. And if you're interested in the references, they're on the last slide. Thank you.